Welcome to part two of our briefing series, Scientific Foundations for a Response to Addiction. Thank you for joining us today as we convene leading scientists to explore the pivotal role that science plays in understanding and addressing addiction. I'm Diana Fishbein, otherwise known as Denny. I have spent the last 40 years conducting research on addiction and related issues funded largely by NIH and also ONDCP, SAMHSA, foundations, and other sources. Personally, I've been particularly focused on conditions that influence pathways towards substance use and mental and behavioral health problems in children and youth, because addressing the adversities that contribute and providing positive conditions that foster healthy development and well-being can prevent substance use in the first place. The sponsors of this briefing are the National Prevention Science Coalition to Improve Lives that we founded about 11 years ago, which is the first organization in the United States to systematically translate science to public health policy, and relatedly, the Addiction Science Defense Network. To preserve the funding for research and evidence-based programs that are so critically needed, we formed the ASDN about a year ago for an exchange of information and to provide an action network for individuals and organizations that study, treat, and prevent problems caused by psychoactive substance use and other addictive behaviors. Our network includes research societies, addiction journal editors, practitioner organizations, programs engaged in the training of scientists and clinicians, and other organizations that overlap with the addiction science community, as well as individuals with lived experience. We are advocating in Congress and with federal agencies to preserve funding for the research that is foundational to our nation's response to substance use crises. We are also urging that the federal government reinstate and scale up supports for states and communities to be able to reliably deliver needed programs and services. And finally, we're informing the public so that they too understand the importance of urging their representatives to preserve this life-saving infrastructure. Addiction remains a profound public health challenge, touching every community across the U.S. While the costs are economic and social and personal, they're immense, but so too are the opportunities before us. Groundbreaking research continues to reveal the complex biological, psychological, and social factors driving substance use disorders. Through continued federal leadership and investments, especially by institutes such as the NIH, we're making significant strides towards developing effective treatments and determining predictors of substance use and dependence, how different people are likely to respond to treatment, and what the risk factors are for relapse. And now I would like to introduce to you Congressman Bill Foster. He is a scientist and a businessman representing the 11th District of Illinois, a position he's held since 2013. He also represents the 14th Congressional District of Illinois from 2008 to 2011. He is the only PhD physicist in Congress, and he was a member of the team that discovered the top quark, the heaviest known form of matter. He serves on the House Financial Services Committee and is the ranking member of the Subcommittee on Financial Institutions and Monetary Policy. I know this is like a mouthful. Previously, he served on the House Science, Space, and Technology Committee, where he fought for evidence-based policies and forward-thinking approaches to some of our nation's most pressing issues, including climate change and energy innovation. The congressman continues to be a champion for sustained federal funding for scientific research. And now the floor is yours.
Am I audible and visible here? Okay. Well, thank you all, and thank you all for your interest and involvement in this. As when I left science 15 or so years ago, and entered politics, one thing I was not ready for with the stories I heard from my constituents about families that have suffered the loss of a loved one from addiction. And one of the real changes in the last 15 years is that people have started to see addiction as less of a moral failing and more as a diagnosable, treatable medical condition. And I was struck. Obviously a big data guy, and I was very struck with the quite often reproduced and valid observation that if you simply are trying to treat addiction with only counseling, your probability of success is in the single digits. And if you add to counseling medically assisted treatment, you get an order of magnitude increase in the probability of success. And that's a big number, and it's one we should pay attention to. I'm also proud as a representative of two national labs, one of which is Argonne National Lab, where in the suburbs of Chicago, where a Nobel Prize was won with research into what are called G protein coupled receptors. Now, this is the molecule that when the opioid eventually makes it to the cell, you have to get the signal inside the cell, and the receptor that does that is a G coupled protein receptor. And it is this fundamental understanding. These now can be imaged in action by the facilities, in this case at the Department of Energy. But the data and the implications of them are analyzed by researchers funded by many sources in the federal government, including the NIH and in large amount. And so the other thing that I find tremendously exciting is that the fact that there is sort of a worldwide race going on to catalog all the receptors in our body and what they do, and all the complex feedback loops that make our body, try to revert to its normal, healthy state when there's an upset. And so you see, as we understand more and more about each of these receptors, you can see that the science is becoming visible. One of the big changes actually in the public perception just in the last few years is the visibility and the visible success of the GLP-1s, the Ozempics, and these drugs that were developed for diabetes, and because we keep our eyes open scientifically, realize that they were effective for obesity. They are effective. There's a 20% drop in all-cause mortality. But one of the interesting things to me is that, it hits, I think, 5 or 6 different receptors in your body, a couple of which are in your brain, and it apparently has a significant effect on the addictive response. Now, you're seeing really pretty impressive clinical results now that show that alcoholism is suppressed significantly by the GLP-1s. And so it's another clue to follow to understand just how we identify the fundamental mechanisms of addiction. And then once you understand them, you can say, how do we suppress them? How do we move all the body's feedback set points to their proper functioning? And there are just tremendous other tools that are coming in. I mean, you can look on the brain scans of people and see the difference between an addict's and a non-addict's brain. And then when they go on different medically assisted and other treatments, you can see them reverting towards normal. And so it's just, incredible. And this is something that the public is really hungry to hear about. One of the best received events that we ever had for the people back in my district was a special event on the science of addiction, where we had members of federal research teams and as local doctors. And what we found is that the people who appreciated that the most were the family members who had someone who had suffered or maybe someone they had lost to addiction. And just the thought that yes, we are understanding the science and that there is hope going forward, that addiction is not a one-way street that you'll never come back from, is something that just provided real hope and was very warmly received. And this is really an example of government doing what people expect it to, which is to do real things to improve your lives. And it's going to be a mixture of long-term fundamental research, like figuring out what's going on with G protein coupled receptors. And there are and detailed real world clinical trials to see what really works and get evidence-based things that will allow me to go to Congress and say, let's turn up the funding for programs that I can prove work. And so the whole spectrum of effort there is important to support. And I just want to thank all of you for being part of that. And I wish I had time to actually listen to the technical presentations here because, it's fascinating. The rate of progress is impressive and the good that it will do for people in the United States and all of humanity when we hit the finish line and can really, put addictive behavior into the box of things that we can control and eventually get rid of. That will be a moment to celebrate, and it's not 2 lifetimes away. So thank you all.
That was so excellent and just the perfect segue to this briefing. You introduced exactly the kind of science that we're going to be talking about today. And since you're going to miss it, you will get a recording. So you can watch it along with others that weren't able to make it in real time as well. Thank you so very much for your time, Congressman.
No problem at all. Happy to help. You guys.
Thank you. You take good care.
Okay.
Okay. Okay. All right. Moving on. I did want to mention the next briefing that we're having. I believe it will be on February 25th because this is a series. We're having 6 or 7 of these webinars. I believe it will be on February 25th. If you can snap this picture of the QR code real quick, you can keep in touch there. You'll also receive a announcement in case you're able to join. That one will be on connecting addiction science to public health policy. So for today, here are our speakers. Today's session will highlight ongoing and future scientific advances in addiction research, and we're really honored to be joined by distinguished experts whose work is really at the forefront of innovating new therapies and approaches. The panelists bring significant expertise spanning a whole range of disciplines: pharmacotherapy, neuroimaging, computational modeling, clinical perspectives, and they're going to offer a real comprehensive view into the science that is shaping our response to addiction. And I just want to ask that as you're listening to these presentations, keep in mind the critical importance of translating these scientific discoveries into real-world solutions that will guide clinical care and ultimately improve the lives of those who are suffering from addiction. So super quick, feel free to message each other in the chat, but we can't track it. We do look later to see if there are any important links or resources. It's just it goes by too quickly and we're not able to keep track of it. But if you have questions for the speakers, put them in the Q&A box and we will respond to them as soon as possible. And you will automatically receive a link to the briefing and resources and slides and so forth a few days after the briefing. So from there, I'm going to allow Vani to access the platform, and I'm going to introduce her. This is Vani. Oh, there you go. Vani Pariyadath. She most recently served as the chief of the Behavioral and Cognitive Neuroscience branch at the National Institute on Drug Abuse at NIH. In that role, she led a multidisciplinary team overseeing research grant portfolio, including large-scale initiatives such as the NIH Healthy Brain and Child Development Study. So she's going to kick us off today.
Thank you, Denny. Thank you for the introduction and for coordinating this whole briefing today. As Denny noted, I was at the NIH. I was at the National Institute on Drug Abuse until mid last year. So today I'm hoping to provide a high-level picture of NIH's interests, questions that have been looked at the NIH recently, and the progress that's been made at NIDA and NIAAA specifically. So this is really meant to be just a broad strokes picture. It's not going to be a comprehensive description at all, but you will have a chance to hear some specific examples from the speakers after me. So NIH has used a variety of approaches to study addiction, to study risks and protective factors, and use that towards arriving at interventions for treatment, for prevention. And one valuable resource that we've had is various cohort studies that involve populations of interest. So for example, the PAT study that's focused on tobacco and health, and another one shown here is the Monitoring the Future cohort study that's been tracking a group of children and teenagers for years. And cohorts like this, they give us glimpses into population behavior. Monitoring the Future, for example, here shows us how overdose death rates have changed over time, and it allows us ways of looking at emerging trends. These kinds of studies have also been supplemented by projects like the Adolescent Brain Cognitive Development, or ABCD study, which also you'll hear about more in a future talk. And this project combines these population approaches with collecting neuroimaging data, brain data, behavioral data, giving us glimpses into some of the underlying behaviors of interest with substance use, but also even for behaviors like screen use, that's shown here as an example. These cohort projects, they also give us a way to look at potential systemic factors in substance use and substance use disorder. They give us a way to look at the impact of, for example, policies, state-level programs on brains, on behavior, on mental health. And in addition to these population studies, we also seen the NIH support a variety of laboratory-based approaches to study substance use behavior and underlying brain function. So this might be involving humans or animal models. And I do want to note here that we typically talk about science moving from the lab from understanding the problem to developing interventions to then moving that out into the real world. But in actuality, science doesn't make progress linearly quite like that. And you heard from Congressman Foster the example of GLP-1 receptor agonists or medications like Ozempic that was developed to treat diabetes. But once it was out there in the real world, it was observed from population studies, from real world insights that even people with substance use disorders seem to be benefiting from drugs like Ozempic towards curbing substance use behavior. And once this was noted, this was taken back into the lab through NIH-supported studies to look at the underlying mechanisms and what might be driving these changes in real-world behavior. And this direction of going from using clinical insights towards the lab has been extremely productive at the NIH. So, for example, in substance use disorders, researchers have used insights about risk and protection due to environmental factors, due to genetics that have been observed in clinical populations, taken that to the lab and unpacked it further using animal models, where they're able to look at very specific behaviors that might underlie the complexity of substance use disorders, and then look at underlying brain function, use these to bring new insights that could then be taken to help the people with substance use disorders. I want to note that while we've learned a lot from these population studies, from these individual level studies, there's a lot that remains poorly understood even now. So for example, here we're looking at the overlap between substance use disorder and mental health, various mental health conditions. And while this has been well known for a while, it's poorly understood, why there's so much overlap in terms of symptoms, in terms of the underlying risk and protective factors. And in recent years, NIH was moving towards understanding this further. One reason it's of great importance is because when you look at treatment rates in substance use and mental health disorder, you see that there's a massive treatment gap at this point. So, in fact, if you're looking at this data, to simplify it, really the takeaway is that the vast majority of people with substance use and mental health conditions are not receiving adequate treatment. And these numbers start to look even more worrisome as you look at it across different communities. So if you're looking at a breakdown based on racial and ethnic groups, you see that there are persistent disparities in how different communities are receiving treatment. NIH had recognized these disparities and was looking to make progress towards understanding various social drivers of health. So to try to understand how various systemic factors intersect with the individual. So for example, shown here is the NIDA Racial Equity Initiative that set up a collaborative of research projects to look at questions related to such social drivers of health. So this was a combination of a large number of project teams that were using anything from laboratory-based approaches to more applied methods, examining services, interventions. And they were using this in a way that carefully defined the various social factors driving disparities. And they did this in a way that partnered with the communities that were impacted. This kind of a holistic approach was necessary to really get at potential real-world solutions. Now, in the last year, we have a host of new challenges that are affecting our ability to arrive at real-world solutions. So shown here are terms that have been removed from funded grants at the NIH over the last year, and you can see that they relate to health disparities, for example. This requires us to have new conversations about the implications and discuss ways to move forward that would allow science to progress through a free exchange of ideas again. I want to finish by bringing back this landscape that I showed earlier, showing us where addiction stands right now. Like I said, there are lots of important problems that still need answers, and now is not the time to slow down research. And now is absolutely not the time to ignore the communities that have been disproportionately impacted by substance use problems. Thank you.
Thank you so much, Vani. That was excellent. It gives us a great overview into what we're going to be discussing today. And now we have Hetty Kober. Dr. Kober is a professor of psychology at the University of California in Berkeley. She also is appointed at the Helen Willis Neuroscience Institute, and until recently, she was associate professor of psychiatry, psychology, and neuroscience at Yale University.
Hi, everyone. Lovely to be here today, and thank you for coming and for inviting me, of course. So I come to you today wearing a few different hats. I'm first of all, a licensed clinical psychologist who treats individuals who suffer from addiction. I'm also a clinical researcher studying treatments for addiction in my lab now at Berkeley. And I was trained also as a cognitive neuroscientist who uses functional magnetic resonance imaging, or fMRI, to study the brains of people with addiction. And finally, I'm a professor. I'm a university professor, and I teach undergraduates about addiction, and I teach clinical graduate students about diagnosis and treatment of addiction. But before I was all of these things, I was actually a little girl who lived in a house with parents who were very heavy smokers. I didn't know it at the time, but they were addicted to cigarettes or had what we now know clinically as tobacco use disorder. And I really found this absolutely disgusting as a child. And so imagine my surprise when I tried my first cigarette in college and discovered that my brain loved exactly what my parents' brains loved, which is nicotine. And I very quickly became addicted and started smoking in many, many different contexts. And then very quickly also tried to quit and relapsed and tried to quit and relapsed. And I really wasn't successful in my quit attempts until I discovered that what really tripped me up every single time was a strong rush of craving that prevented me from continuing my abstinence. And since I'm going to throw the word craving around quite a bit, I'm going to give you my working definition. I also often define craving as a strong desire or wanting. And importantly, this is something that has troubled philosophers for centuries. Over 2,000 years ago, the Buddha said that craving is the cause of all suffering. Baruch Spinoza said it is the very essence of man. Bertrand Russell said that it prompts all human activity. And they were all highlighting something that today we know from epidemiological work is just exceptionally common. Almost all adults report feeling craving for something sometimes. Psychologically, I think of craving as a multidimensional construct that has an affective component, the feeling of craving, but also a cognitive component, which is the thought, I want this, but also expectancies. If I have this, I'll feel better. If I don't have it, I'll feel worse. And of course, a motivational component, which is associated with the impetus to act on these thoughts and feelings. It also has a strong neurobiological basis, which I will talk about in just a few moments. It has long been considered a key process in addiction, and it was added to our diagnostic manual as a diagnostic criterion for addictive disorders in the last edition that was published 13 years ago. And just in case that wasn't quite clear, I'm going to start with a little demonstration. I'm going to ask you to look at these fluffy, fresh chocolate croissants. You can imagine they just came out of the oven. You might look where the glaze goes on these. It's so shiny. You might imagine that the chocolate in it is just really warm. You know, this is how, of course, food commercials work. This is actually my personal favorite. It's a sea-salted chocolate brownie, sea-salted caramel chocolate brownie with some nuts on top. And if you happen to be a participant in my lab, I might hook you to some equipment and I might be able to measure that your heart started beating just a little bit more quickly. Maybe your palms began sweating. We call this Q activity. And I might put you in an fMRI machine and look at your brain activity as it changes when you look at these food images. And I might ask you, did you crave any of those? And if we were in a room together and I could ask you to raise your hand, most people would give it a rating that was higher than not at all. And I would then argue that you had experienced craving for these foods. And of course, everything that food can do, drugs can do better. And so when we bring individuals with addiction to the lab and we show them pictures of their drug of choice, I'm going to show you here pictures of cigarettes, cocaine, methamphetamine, alcohol, and marijuana. Hopefully I'm not activating anybody. Those individuals report to us that they feel craving. We specifically call this Q-induced drug craving. And the ability to do this in an experimental setting has been known for 35 years at least. And today I'm going to make 3 important points about this kind of research in the time that I have left, which is craving is a key factor in addiction. It predicts drug use and has clinical implications for clinicians. We can teach individuals with addiction to regulate or reduce their craving, and this reduces substance use. And we can parlay this into digital interventions for individuals who don't otherwise have access to treatment or as an adjunct to existing interventions. And we can also identify the neural mechanisms of craving, which has clinical implications for diagnosis and for the prediction of treatment outcomes. And hopefully all of this will allow you to understand how continued support for addiction science is crucial. So the first question is, does drug craving predict drug use? And here I'm going to show you not a single study, but a meta-analysis, which is a study of studies. It allows us to measure the consistency and strength of the evidence across the entire literature, all of the studies that have ever been published. And here we found studies that at time 1 measured some form of craving, and at time 2, which could be minutes, but also months and years later, some measure of drug use or relapse. And we identified here data that represents 51,788 drug users. This is a very large sample. What we found, in short, is that craving absolutely predicts relapse in the future. This was significant across every analysis in our data set. And to give you a sense of the strength of the findings, some effects had an odds ratio of 3.1. And what does that mean? It means that 1 unit increase in craving more than triples the odds of likelihood of drug use or relapse. And this has immediate clinical implications, which means implications for the treatment of addiction, that craving should be measured frequently during treatment. It's a meaningful indicator of relapse risk in our patients. And we can perhaps also use it as a treatment target, which led us to ask, can we actually teach individuals with addiction to reduce their craving? And we developed a set of tasks, a task to do this, the regulation of craving task. And over what you can see here below as all of the references from literally my entire career, we taught individuals with addiction to use specific strategies and found that they can indeed reduce their craving. And this includes cognitive strategies as well as mindfulness strategies. And we also found that when these people used these strategies to reduce craving, it also modulated their brain activity in regions that are associated with craving, which is incredibly important for understanding the biological mechanism underlying this important effect. We also then started to systematically train individuals to do this in the context of regulation of craving-based digital treatments, and we found that they did reduce drug use, and we think that this is ideal as an adjunct intervention for medication treatment for those without access to other forms of treatment. And when we think about identifying the neural mechanisms underlying craving, here we're using, again, fMRI data that we collected when I was still at Yale. We recruited individuals with tobacco use disorder, cigarette smokers, individuals with alcohol use disorder, individuals with cocaine use disorder. This is crack cocaine in particular. We showed them the images of their drug of choice. We measured their craving, and then we applied machine learning, an advanced statistical technique or a computational technique to these data. And what we were able to identify is a pattern that we now call the neural craving signature, or the NCS, that spanned the entire brain and allowed us to identify regions with positive weights that predicted higher craving and regions with negative weights that predicted lower craving in these individuals. And again, we immediately were able to find some clinical implications here, which is the NCS, in fact, is diagnostic of the addictive condition itself. We were able to classify individuals with and without addictions with 82% accuracy, which is actually higher than most groups of clinicians, 60% specificity and 97% sensitivity. And you should know that these numbers are actually quite good. For example, mammograms have about 80% sensitivity in detecting breast cancer, and we use that in clinical practice every day. Sensitivity here being the probability of a positive test result if the result is actually positive. This suggests that the NCS can serve as a diagnostic brain marker for this disorder. Importantly, more recently, we applied the NCS to individuals who had to the brain patterns of individuals who had just completed treatment for their alcohol use disorder. This is very recent work, and we found that the NCS also predicted relapse to alcohol in these individuals as far as 3 months in the future, suggesting that the NCS can also serve as a prognostic brain marker for relapse. And so I presented to you here 3 different lines of work on craving, hopefully as a good illustration of how we use research funds to advance treatments for addiction and substance use disorders, and hopefully to reduce the suffering of people with addiction and give them, develop better treatments for them and allow them to build a better life going forward. And I will end with a moment of gratitude to the National Institute of Health that funded literally all of this work. These are my current grants, but there's many, many grants over the last 20 years that have funded this work. And I also wanted to highlight the training grants that we received primarily from the NIH, from trainees in my lab who are all studying or have studied to become addiction researchers and to continue to investigate these important questions. And I also thank you all for listening.
That is a tremendously exciting body of work, Hattie. Thank you so much for presenting it. When I rewatch it, I'm going to put it on slow-mo so I can get every word. Okay, Hugh Garavan. Dr. Garavan is a professor in the Department of Psychiatry at the University of Vermont, and he was the associate professor in psychology at Trinity College, Dublin, prior to Vermont in 2011. Hugh, take it away.
Great, thank you so much. I hope you can all hear me. Thanks everybody for coming. I see that there's like a sizable number of people here, so I really appreciate you taking time out of your busy days to come and attend. So my title here is very grandiose. I'm not going to actually explain what are the causes and consequences of substance abuse, but I'm just going to flag that these are some of the kind of really critical questions that motivate a lot of our research. We don't have deep understandings into the causes or indeed into the consequences of use. You can imagine there's probably multiple causes, genetics, environment, access, peer influences, brain function, and so on and so forth. So addressing any of these questions, including trying to address the consequence of use, these are big, big research endeavors. You'll get a flavor of all that's happening today in these different domains. These are critical, important questions, as has been made clear already in the presenters. So what I want to do, and I guess, we're all here today to try to reinforce the take-home message that this is critical, this is important, this should be protected. Okay, What I want to do is kind of just give you an overview into two rather large endeavors to get at some of these questions about causes and consequences. The first is a project called Enigma Addiction. Basically, what this is doing is pooling data like Hetty just showed, brain imaging data of individuals with substance dependence, individuals without pooling them together, getting good characterization of all of those individuals, trying to understand what might be, in this case, specifically brain correlates of substance use. Second study I want to talk about, this has already been mentioned earlier, the Adolescent Brain Cognitive Development Study. This is a very large longitudinal study. We're following about 12,000 kids from age 9. The idea was to have kind of very rich assessments of these kids prior to any substance use, follow for 10 years, hopefully for 20, 30, 40 years, see what sort of developmental trajectories they have. Some will develop mental health issues, some will develop substance dependence problems. Is there anything in these rich data we can find that will give us an insight into who was most vulnerable, who was most at risk for having one sort of outcome versus the other. And indeed, if they start using drugs heavily, what sort of changes, mental health, cognitive, social, personality, brain might arise with that substance use. Okay, so let's get into just a couple of examples, just kind of give you a feel, give you a flavor for the sort of research that we do. So let's start with the Enigma project. So we're interested in brain markers of dependence. How do we do this? So brain imaging, you're probably all kind of familiar with these sorts of brain images. We can acquire these very, very quickly, just a few minutes in a participant, get these nice high resolution pictures of somebody's brain. We can do some fancy image analysis, strip away the skull. We can zoom in on the brain region itself, and we can actually trace the gray matter. So the gray matter that's around the edge of the brain, we can trace its inner boundary, its outer boundary, and we can quantify it. So we can get good measures of literally how much gray matter, how thick the cortices are in any one individual. And then, of course, we can say, okay, well, with that type of brain measure, we can compare cases and controls. We can look at individuals with alcohol use disorder, compare them to match controls and see other differences. And so, for example, here we'll see that there are differences. You get smaller gray matter, thinner cortices in individuals with alcohol dependence than those without. And you'll see what we're just plotting here is that effect, but as a function of age. So you'll see the effect gets bigger with age. And, this could be, it's plausibly related to the fact that this is an exposure effect. The more exposure, the more years of use, whatever potential toxic or detrimental effects the alcohol use has, it accumulates over a lifetime. Interestingly, though, if you look at this at the younger age, so 18, 20, 22, 24, those differences aren't significant. We don't see a group difference. We don't see an alcohol-related reduction in gray matter volume in those younger individuals. But we do see differences in another brain measure. So what you're seeing here right now is essentially those same data from before. So this again is looking at gray matter volume, gray matter thickness as a function of age. And this sort of binning ages across the lifespan. And you'll see there in the white area, this is where there's no group difference. And in the shaded area, this is where group differences emerge. So you get this difference in the older individuals with alcohol dependence. A different measure, I won't get into the details, but it essentially looks at not just how thick the cortices are, but sort of the patterns of cortical thickness across the brain. It shows an effect at the younger age. Here in the individuals who with alcohol dependence who are younger, they show a difference in this measure. And actually that goes away as they age. And critically, what I think makes this kind of most interesting, and the reason I'm telling you about this is it speaks to this issue of causes versus consequences. If we look at another group of individuals who are 19, they're not alcohol dependent, but they're drinking heavily, we see the same effect. No difference in cortical thickness, but this structural covariance measure is different. And really, really interesting, those same kids at age 14, before they started drinking, show the same effect. So I'm not saying that this is necessarily, a predictive, perfect biomarker, but the point is just to convey to you that with sufficiently large samples, with the ability to look at age-related effects, with longitudinal studies, we can start to get an insight into brain differences that perhaps arise with use, and then maybe also some brain differences that might give us an insight into vulnerabilities for use. What is it about these kids? What it is about their brain organization that seems to predict future substance use? These are the sort of questions that we are really, really keen to interrogate as we're trying to understand substance dependence. Another question that's very much to the fore of people's mind is cannabis use. So again, younger kids, altered brain development. Is there altered brain development associated with cannabis use? There are, and without getting into the details, we can see effects in terms of, brain regions with smaller gray matter volume. This is in kids between ages 14 and 19. This is kids between ages 19 and 23. And these are longitudinal studies. So at the beginning, there are no differences. But as the cannabis use develops across these age spans, we start seeing these differences, these accelerated changes in brain development. In the ABCD study, and just a couple of slides, and then we're done and we'll pass on. But just to let you know that this is kind of a very kind of important topic of research for us, because we have such a large and diverse sample, and we've been following them for 9. We're really keen to kind of understand who starts using cannabis. Is there any way we can predict after the fact who is going to start becoming a cannabis user, or maybe in time develop cannabis use dependence? And what sort of differences might emerge as they start using. So this, just to give you a feel, this is the kind of age of first use of cannabis. We have at this stage about 1,500 kids who've started using cannabis. So we can now look at the vertical lines show kind of when we've acquired data. So we can now look at changes that might arise with the cannabis use. And to give you kind of a very, very sneak peek as to what we're seeing, we can look at early initiators. So these are kids who started using cannabis between the ages of 12 and 14. What I'm plotting there is a sort of a mental health measures. So this is measures, the CPCL is a tool for just, getting reports of symptomatology. So internalizing anxiety, depression, or inattention problems, or on the right, there is externalizing the sort of disruptive rule-breaking behaviors. And what we can see that in those kids who start using cannabis between 12 and 14, that those measures start to increase. And we're trying as best we can to control for a whole bunch of other potential factors that might confound it. In those kids who start between 14 and 16, we're starting to see something similar. So, of course, one of the key questions here is what changes arise? Are the changes different depending on what age or how much you use? Are there differences depending on other factors, other sociodemographics, other psychological factors? These are all the sorts of questions that we're very keen to address. We can also do the same sort of logic and look at brain development. So, for example, again, those early initiators, the kids who start using between 12 and 14, and on the right there, I should have explained, so the green is kind of a measure in our participants who have not started using cannabis, and red are the ones who do start using. And you see that the sort of the typical normative pattern of cortical thinning is accelerated, increased loss in gray matter in those kids who started using cannabis. Similar effect in the hippocampus. We want to be very careful. We want to not jump to any kind of quick and easy causal conclusions that the cannabis is causing this effect, but we really want to understand what might be driving these effects. How is it related to cannabis? What sort of guidance can this give us in understanding the condition? Okay, I will finish up. I just want to thank my colleagues who've done the bulk of the work. The big picture here for you, and this is just kind of a snapshot of a couple of research questions, is that there's loads of these really important questions. I'm sure many of you in the audience here have your own questions that you'd love to get answered. To do this, we really need, unfortunately, big investment, large samples, followed longitudinally, so that we can really understand what's happening and understand all the multiple influences that might affect something like brain development. Significant progress can be made, is being made in understanding cause and consequence of substance use, but we really do need large funded research to make it happen. And with that, I will thank you and hand you over to the next speaker.
Thank you so much, Hugh. That was phenomenal. Incredibly important research. It gives us clues into where, how, and when to intervene, and what kinds of social environments impact the brain at those critical intersections throughout development, so that we can prevent substance use. So now, Martin Paulus. Dr. Paulus is Scientific Director and President of the Laureate Institute for Brain Research, and holds academic appointments, including Adjunct Professor of Psychiatry at University of California, San Diego, and Professor of Neuroscience at the University of Tulsa.
Thank you very much, Denny, and thank you for giving me the opportunity to talk a little bit about the work that we're doing at the Laureate Institute for Brain Research. This is a work done with a colleague of mine, Leander Figueroa Hall. I'm going to tell you a little bit of a slightly different story here about how we're trying new techniques and technologies to unlock what's going on in the brain of people with substance use disorder. So, we've been, and you've heard quite a bit about this already before, we've been focusing very much on what's going on in the neurons and the nerve cells of the brain. And of course, they are extremely important to compute all the things that we are experiencing, the thoughts we're having, the feelings we're experiencing, the actions we're taking. But actually, if you think about it, if you look into the brain, they're only half of the story because the neurons are only one part of the brain. There's a whole set of other nerve cells in the brain that are important to set the environment of what these nerve cells are computing. And we've been particularly interested in what is going on with these cells, and could they contribute to what's happening when you have a diagnosis such as opioid use disorder, which really is a complex disease that undergoes lots of physiological changes and goes way beyond just taking a particular drug. It really changes fundamentally the body of the person. So we've been trying to find out, can we unlock the secrets of what these other cells tell us about people with opioid use disorder? And as it said, that one of the cells we focused particularly on is called the astrocyte. The astrocyte can be thought of as sort of the brain support system. It has a lot of different functions, and we're still, based on some basic neuroscience, trying to understand all of its functions. But it controls inflammation, it supports brain health, it regulates the environment that the neurons live in. And it is extremely important for how nerve cells communicate one another. And also it's important for how signals go beyond the brain to the body. So we've been focusing on this, on these astrocytes for quite some time now. But the problem, of course, is that we're facing is you can't just stick a needle into somebody's brain. I mean, under extreme circumstances, of course, you need to, but you wouldn't want to in the day-to-day environment. But something that has really developed over the last 10 years that has made us so excited is what's called extracellular vesicles. So let me tell you a little about these little bubbles. These are little vesicles that are exchanged between cells in the body. The way to think about it, they're all like little message boxes. And the message boxes come from everywhere in the body, including these astrocytes. And what we've been able to do with the help of lots of other previously developed tools and techniques, we're able to isolate these extracellular vesicles from the blood. So what we now have, we have an ability to actually see what's going on in the brain from actually getting a blood sample. So this allows us to essentially do what we call a blood-based brain biopsy. We don't have to stick anything in the brain. We just basically take a standard draw of blood. We do a really sophisticated set of biochemical analyses, and then on top of that, we do bioinformatic analyses, and then we are looking what is actually inside those little vesicles. And so what we found, which is actually quite exciting, and we've been doing several cohorts now where we see similar things. We're looking in this particular study, we look particularly at proteins. Proteins are important for lots of different things. They, of course, they're built, what the body is made of, they're enzymes, but they also control what we know as inflammation. We all know when we have inflammation, you know, your skin is red, it hurts, it swells. And inflammation can happen all over the body. It can happen in your organs, it can happen in your brain. And what we found when we actually looked at these astrocyte vesicles that we extracted and enriched and identified in the blood, that there was a particular pattern of proteins that was upregulated and other patterns that were downregulated. So now you might ask, okay, what kind of proteins were upregulated and downregulated? Well, what we found is that the ones that were upregulated were primarily involved in chronic immune stress regulation. These were proteins that were important for inflammatory processes. So essentially, we saw that inflammatory proteins were increased, and the regulators that normally keeps inflammation in check were decreased. And here's just one example of two types of proteins that we found upregulated or downregulated respectively. This is just to kind of illustrate the point that what we were able to do by looking into these vesicles that were coming from the brain, but that we got from the blood, we show that people with opiate use disorder have actually an inflammatory process going on in their brain. And why would that be important? Well, it changes the way you even think about addiction. Now, and we've heard from the congressman already that we've made significant advances in moving away from thinking of addiction as just a disease of willpower. Lots of work has been done to implicate molecules such as dopamine in the addictive process, but now we're even going further. I think we can begin to conceptualize addiction as really a whole body systems disorder that involves neuroimmune regulatory process. That this is not unlike what we're seeing with other complex immunological disorder like multiple sclerosis or other neuroimmune disorders. That really what we're seeing is that the brain is so fundamentally changed that we need to then look at how can we treat the brain differently. So, the finding that we're having with these opioid use disorders has really direct consequences for how we think about future treatments. Clearly, if inflammation is out of control in these people, then what we need to do, we need to try to bring it back into control. And what's interesting is for some of these proteins that we discovered in these extracellular vesicles coming from the brain, there are already targets for medications. We heard earlier about how we discovered that GLP-1 drugs, the Ozempics of the world, are useful for many different things. Well, we have a number of immune modulators. And wouldn't it be fantastic if we discovered that these immune modulators could also help people to overcome opioid use disorder? So I think that this is really an example where we can see how this can open up a whole new line of research. It also helps us to develop potential blood tests to better differentiate between different types of opiate use disorder. Just like many other brain diseases, we call people with depression having depression, but there are different causes of depression. There are different causes of opiate use disorder. So can we begin to differentiate subgroups of people based on this approach? And finally, can we go beyond asking questions, have you used today, or how bad are your cravings? As important as that is, and as Eddie pointed out, that is important. But can we go beyond that and get a blood test and tell how strongly addicted the body still is and how much medication the body might need to overcome addiction? So I think that I wanted to kind of convey the enthusiasm that I have about this approach because of a number of different reasons. First, it's a minimal invasive approach. We could potentially use this just from a simple blood draw and discover what's going on in the brain. It's discovery based. Without basic science, we could not do this work. We were able to use proteomics and bioinformatics to basically identify these proteins. It gets us to a measurable level. One of the things about psychiatry and addiction medicine in general is that unlike cancer and other medical areas, we still have a very difficult time having quantifiable biomarkers. And with this approach, we can. And finally, it is actionable. It leads us to something directly that we can treat people with. So I think we're entering a new era for addiction medicine. And I really think that this could not have been possible without the support that we're getting from the National Institutes and other organizations that support research in addiction. Thank you very much.
I have to say that you all are getting literally a sneak peek at the most exciting advancements in the field from these speakers. And now we have Dr. Henry, aka Hank Kranzler. He is the Carl E. Rickles Professor of Psychiatry and Director of the Center for Studies of Addiction at the University of Pennsylvania, Perelman School of Medicine. Hank, are you there?
Yes, I am. I'm sorry.
Oh, good.
I was muted.
Nope. Okay. You're you need to go to slideshow.
Yep.
Okay.
Okay. Starting here.
There you go.
It's a pleasure to be here. This has been a very interesting series of talks. And I am going to focus not so much on the data, because there's just too much data to present on the pharmacotherapy of addiction, but I'm just going to talk about the current landscape and where we may go from here. And I'm going to cover, before I do, I work with industry, and this is a disclosure of that work. The vast majority of individuals with a substance use disorder never get treatment. The most common form of recovery is spontaneous recovery, if you will. People making efforts on their own to stop smoking, as Hedy talked about. But certainly the same is true for drinking. There are FDA-approved medications for alcohol, tobacco, and opioid use, not for cocaine or cannabis. So it's the those three disorders are the ones I'm going to focus on to talk about medications development. But they're grossly underutilized. And we need research to optimize the use of the available medications and to identify and test novel ones. And it's a long process to develop a medication for any indication, and particularly to get FDA approval. In the US, there are 4 drugs, actually it's 3 different drugs, but 4 different formulations for treating alcohol use disorder. The first, disulfiram or Antabuse, was approved 65 years ago. Naltrexone about 30 years ago, acamprosate 20 years ago, and extended release naltrexone, the last drug to be approved for treating alcohol use disorder, is now 20 years old. We really need more medications. The effect sizes, the effects that these drugs have is small. It's significantly better than placebo, and that's why the FDA has approved them. But still the effects are small. Similarly, for nicotine, the most recent drug, varenicline, was approved 20 years ago. Nicotine replacement started more like 40 years ago, and different formulations of nicotine replacement have been approved. But again, these are comparatively small effect sizes. Bupropion, the antidepressant, was repurposed in 1997 for smoking cessation. And as I mentioned, varenicline, which is probably the most efficacious medication for treating smoking, was approved 20 years ago. And then lastly, for opioids, and I want to just give you an overall lay of the land here in terms of what drugs are FDA approved. Methadone has been around for a long time, and it works. It works extremely well. These are larger effect sizes. These drugs like methadone and buprenorphine in particular have been shown to save lives. And naltrexone was approved as an oral formulation for treating opioid use disorder before it was approved for alcohol use disorder. Buprenorphine now is available as is naltrexone in both oral and extended release injectable formulations. So this is where we are. There are clearly drugs that are efficacious, some particularly effective, like methadone and buprenorphine and life-saving, others with smaller effect sizes, but still robust effects like varenicline. And probably the effect sizes for alcohol use disorder are the smallest as a group. There are novel medications in development for treating substance use disorders. These include psychedelics. There's an early literature on using LSD for treating alcohol use disorder that supports the use of psychedelics for that indication. There's also initial evidence in a placebo-controlled trial of psilocybin for treating alcohol use disorder. There is a literature on smoking cessation as well. Perhaps the most promising group of drugs, because the regulatory burden is not as great as it is with psychedelics, are the GLP-1 agonists. The congressman mentioned them. Others have mentioned them in their presentations today. And these are a very interesting class of drugs. There is a robust preclinical literature supporting their use in reducing drinking in animal models and initial evidence of clinical efficacy. There are quasi-experimental designs using electronic health records data, but only a limited number of clinical trials, which are the gold standard in evaluating medication effects, placebo-controlled trials. And there's one that's particularly compelling with semaglutide, which is marketed as Ozempic or Wegovy, for treating patients with AUD. And there are probably 10 studies that are listed on clinicaltrials.gov of semaglutide in particular, but some other GLP-1 agonists for treating alcohol use disorder. So just to reiterate some points and then talk about where I think we're going and why it's important that NIH play a key role here. Substance use disorders are common in the US general population. They're probably the overall prevalence exceeds 25% in alcohol and drug use disorders in terms of past year or current substance use disorder diagnoses. There are clearly efficacious psychosocial treatments and self-help group participation, which have been discussed briefly today. And these provide a foundation for behavior change, but they too are underutilized. Adding efficacious medications can help augment the effects of psychosocial treatments and reducing substance use or promoting abstinence. But as I've mentioned, these are seriously underutilized. In contrast, GLP-1 agonists, which seem to be on everybody's radar screen these days, are now being used by about 12% of Americans. That's roughly 1 in 8, and that's a doubling over about the past 18 months. So the rate of increase is dramatic. These are drugs that are highly efficacious in reducing hemoglobin A1C, a measure of diabetes risk or a measure of diabetes, and in reducing weight in people with overweight or obesity. And they also seem to reduce substance use, both alcohol use in clinical trials, smoking in a smaller literature, and they're being tested now for opioid use disorder as well. There's an important message here. the discovery of the hormone glucagon-like protein 1 and medications that imitate its actions were largely academic. Rockefeller University, Mass General, and it was only the translation to a medication that was the purview of the pharmaceutical industry. This really reflects a very successful collaborative effort between NIH-funded investigators and industry to bring a new class of drugs to the clinic. And the inadequacy of NIH funding threatens a number of elements of this successful pipeline. There will be a decreased pool of novel medications stemming from the basic and early clinical research that NIH and NSF and the VA fund. There'll also be, importantly, a decreased pool of investigators to conduct this research. And this will result in a greater dependence on industry, which will select candidates for study based on financial rather than public health considerations. Now, don't get me wrong, I don't mean to vilify industry. I work closely with industry. They play a key role in drug development and certainly in dissemination of products once they're approved. But the primary motive of the industry is profit, not public health. The primary motive of the NIH, NSF, and the VA is the public health. And I think this is a key consideration for this audience, and I commend it to you. And with that, I will stop my presentation.
Thank you so much, Hank. That was great. It gave us a really good, broad overview of all that the science has produced in terms of medications that are being used and how critical that science
I
is.
wish I could stop sharing here.
Oh,
But
I think I can.
yeah, you
I
can
think
stop
I
sharing.
can take you down. No, that didn't do it. Well, you just mess with it while we talk. Anyway,
Okay,
I
go
thought that
ahead.
was terrific conversation, and I hope that the takeaway from all of these talks has been what this research has led to is just tremendous advancements and innovations in our ability to respond effectively to substance use disorders, as so many people in this country are suffering. We need more of this independently conducted research that's funded largely by the federal government. So I want to just, we're going to do questions and answers now, but I want to remind you that we have more briefings coming up. The next one is on the implications of addiction science for public health policy, we believe on the 25th. So please, check out our website. We also, after that, will focus entirely on prevention, which is the most dear to my heart, and hopefully many of you as well. So you haven't figured that out yet, huh, Hank?
It's still up there? I thought I did.
Yes. Yes.
It's amazing. I'm going to.
So yeah, it's okay. Just keep fussing with it. And can everybody else get on their videos, please? And we'll ask questions and so forth. Thank you so much. And at home, in the office, wherever you are, you can there we go. I was going to say you can minimize the screen. Okay. So great. So, Vani, why don't you start with the first general question?
Sure. So this is a question for the panel. Everyone's welcome to jump in. What do you see as the biggest unanswered questions in addiction science that need the most attention right now?
I can start.
Okay.
Go ahead.
And it's a little bit biased because it's a project that Martin and I once upon a time wanted to get funded. There's a number of questions in the chat. People are always interested about, well, if I stop using, do things recover? Can I go, is this fixable? I mean, it's such kind of an important question. We don't have, I don't believe, a large longitudinal study of people entering treatment. And if we did, we'd be able to, characterize those people as they enter treatment. That might give us insights when they eventually successfully quit or they relapse. What is it about those who, were successfully able to stick with it? What is it about those who were able to quit? What's the time course of recovery? Is there a time course of recovery? Do some brain regions, for example, do some cognitive abilities recover? Do some of them not recover? Do some recover in a week? Do some require 10 years? That would be so useful to have. And it complicated because there's so much relapse and it's a complicated population to work with. But I would love if we had that study ongoing. I think there's a lot of valuable questions we could ask.
Yeah, I just to kind of second what you were just saying. I said I think we need to think of addiction or substance use disorder as more than just taking the drug, and it changes the brain really in very many fundamental ways. And just like any other disease that we need to understand chronically, we need to understand the chronic progression or recovery of addiction, because it may change the way we need to treat people. You have to base your treatment on the biological process of what's actually going on. If you have a pneumonia and you have a viral pneumonia or you have a bacterial pneumonia, it changes completely how you treat that condition. We need to get to that level, and I do agree with you having a longitudinal data sets of people who unfortunately will go through relapse recurrences and periods of abstinence, I think is going to be critically important.
Yeah. Anybody else?
Yeah, I think the big issues are what causes addiction. And I don't mean in broad terms, but I mean in granular terms, because that really sets up how we intervene, how we prevent, and how we intervene. I don't think until we know the relative impact of different individual factors, environmental factors, and genetic factors, and the gene by environment effects in a fairly detailed way, I don't think we're going to have a good handle on how to prevent and treat substance use disorder. And I don't mean that to suggest that to be pessimistic, but to be realistic about how daunting a task it really is. And we're certainly making headway, and I think it's paying dividends, but we have ways to go.
Mm-hmm. Yeah, and I'll just say we need not to ignore the social and structural factors that contribute that can be addressed more broadly through public policy. So there are individual level approaches that must continue to be rolled out and advanced and become more precision based, but also to recognize that the social environment and the supports that families have and the ability for children to grow up healthily and thrive and flourish is really critical to avoiding pathways to substance use in the first place. Hattie, did you want to say anything?
I wanted to add a meta comment that I totally agree with everything that was said, and that one of the things that I think is underlying and also is a theme across some of the work that was presented today is that a lot of this work, especially if we think about long-term longitudinal studies, clinical trials, is that we need lots of labs from lots of places to collaborate together to create the kind of data sets that would allow us to really understand with precision and with accuracy and with power the kind of effects that we're looking for. And for that, we really depend on the NIH allowing us to fund these large collaborations, because even if individual researchers sometimes can get a small pocket of money for these large collaborations across labs, we really need the NIH's support.
Yeah, absolutely. I'm going to follow that with another question for you, Hetty. I want to know, because your work is just so exciting, I want to know what you're most excited about. What is it that you found relative to craving and the implications for clinical practice that is most exciting and tantalizing for you moving forward?
Just one.
Yeah, just one. No.
I mean, I think that one of the things from interacting with clinicians, I know that for them to know that actually they can take the kind of varying amounts of craving that they assay from their own patients, and that that is actually indicative of relapse risk, is something that is already being put into practice in EDs, for example, where you can ask individuals who are coming in. That really gives you a prediction of how likely they are to relapse. I think that clinically, we're also really excited about the possibility, certainly when we finish maybe the next cycle of grants, of distributing the digital treatments that we're developing so that, for example, people who have other treatments can use them as adjuncts. For example, one of the things that we're testing is in people who are already in methadone treatment, if they have this additional training and regulation of craving, does that reduce their relapse? We have some evidence suggesting that it does with other substances. For example, regulation of craving in addition to nicotine replacement therapy for smoking. And I'm also really excited about what we might be able to do with predicting treatment outcome and relapse using brain imaging techniques. I think that there are lots of ways to think about that as helpful when people might not have the insight themselves.
Excellent.
May I address, there's a question from Jody about large trials of GLP-1 receptor agonists. There are no published large trials. There are multiple trials underway. We're actually doing 3. Well, actually, 2 of them are large. They're actually registration trials. It's going to be a few years, but the next drug approved for alcohol use disorder, I believe, will be a GLP-1 receptor agonist. And there's a great deal of interest across multiple companies, large and small. And what's, I think, particularly promising about GLP-1 drugs is that primary care doctors and doctors of a variety of, and nurse practitioners and other prescribers are comfortable prescribing these drugs. That's why I did not go into great detail about the psychedelics, because I think it's going to be a niche opportunity for treatment with psychedelics, even when they are approved. And I want to add that GLP-1s are not without side effects. They're not over-the-counter kind of drugs. They probably never will be. You can induce nutritional deficiencies with these drugs because they have such potent effects on appetite. So they have a way to go. They're expensive still. So I don't know how broadly they're going to be available for treating addiction, but they certainly are a promising group of drugs. And hopefully we'll have large trials published in the next couple of years.
Danny, if we have time, I had another question for the panel.
Yes.
To multiple members on the panel, just picking up a thread from Hetty. I was just curious, what you see as insights that weren't possible before these various large-scale studies, these collaborative efforts that we had between different labs or projects like ABCD, but, in general, this new team science approach that we've seen in the last decade or so.
I'm sorry, I missed the question. What was it?
The question was, what kind of insights do you think we just didn't have access to before that shift happened?
Right. I guess I can start if I may.
Sure.
That it's complicated. I think what we're finding is, well, a couple of things that with certainly these big samples that where we can do like really robust, reliable analysis with proper kind of cross-validation, test the effect in one half and replicate another, that a true effect is probably a small effect. So the effects are not, there's no huge, big, whomping brain signature of addiction. It's going to be small and it's going to be subtle. I think we're discovering that for those who revel in stats, higher order statistics are going to be the name of the day. What I mean by that is it's not going to be as simple as drug produces this effect. It's going to be in interacting with a whole bunch of other things. If you have a certain genotype, if you have a certain early adversity, if you have a certain socio-demographics, then, and you might show this type of an effect. But if you have different scores on all those other variables, you might show yet a different effect. So I think it's going to prove to be just complicated. That there will be single, bright signatures of addiction, I think is unlikely. It's going to be much more nuanced and complicated. And I think you can only get at that if you have large samples to enable to do that kind of stratification.
I can answer something related to that from a treatment perspective, which is I think with larger samples, we also are more able to test treatment combinations and multiple treatments both together and against each other to try to get these additive effects. For example, as Martin was pointing out, right, if you could give someone both some medication for opioid use disorder, maybe a psychological intervention, and also something for their inflammation, you can have a combined group like that, and also each treatment individually to try to understand not only the effect of each treatment separately, but also their synergistic effects. And again, that is one of those things that is only possible to do when you have really large samples.
So one of the areas where large samples have been obtained is in meta-analytic GWAS, genome-wide association studies, where we've assembled some samples of in the millions. And that's important because those kinds of genetic effects, common variants, are very small effect. Odds ratios of 1.03. So you need huge samples. And there's an interesting opportunity, I think, in using genetics from the UK Biobank. It also has a very large MRI sample, 100,000, along with genome-wide genotype data, to use, in essence, polygenic scores for the imaging variables as a way of extending those findings to samples with genetic data but no imaging data. And that's something we've been working on and are about to translate, we hope, to the Million Veteran Program.
Excellent. So anything else from the audience? I don't know that we have any more internal questions here. I don't see any. Well, this was really fabulous, and it will go out. It'll be archived in perpetuity, and it will be viewed by many, many others. So we had great audience. Thank you all for hanging in there with us. And again, please join our next briefing. And thanks to the speakers.
Take care. It was a pleasure.
Yes. Take good care, everybody.